Okay, so one of the scientific articles that I'm using for my protein paper is called The Mutational Analysis of the E. coli Dead Box Protein CSDA. And my protein is helicase. The researchers that were in this article are Anne Marie Turner, Sheraton Love, Rebecca Alexander, and Pamela Jones. The research was completed at Wake Forest University and Winston-Salem University, which are both in Winston-Salem, North Carolina. Okay, so in the research paper, they acknowledge that RNA helicases are involved in a variety of cellular processes that require modulation of RNA structure, such as RNA splicing, ribosome biogenesis, translational initiation, mRNA degradation, and cell division. They discuss the fact that E. coli cold shock protein CSDA is a member of the dead box family of ATP-dependent RNA helicase. Okay, in the experiment, they show that CSDA exhibits in vitro ATPase and helicase activities in the presence of short RNA duplexes with either 3-inch or 5-inch extensions at 15 degrees Celsius. They tested a wild-type CSDA, which had no detectable helicase or ATPase activity at 15 degrees Celsius in vitro. They also tested a plasmid encoding the variant CSDA, and it was unable to suppress the impaired growth of the null mutant CSDA at 15 degrees Celsius. And finally, they've tested a plasmid encoding CSDA, which lacks most of the carboxyl terminal extension. It enhanced the growth of the null CSDA at 25 degrees Celsius, but not at 15 degrees Celsius. So that protein had limited vitro activity at 15 degrees Celsius. After testing the results of this experiment support the physiological functions of CSDA as a dead box ATP-dependent RNA helicase at low temperature. This article is relevant to my protein helicase because helicase was first discovered in RNA, the RNA in E. coli in 1976, and this experiment dealt with E. coli.